because humans are the ones giving them antibiotics, but that's the selective pressure. Yep. Every population wants genetic variability. The more different types you have, the more likely some are going to survive as the environment changes. Alrighty. So, how do bacteria replicate? Binary fission, and before two cells can divide, before one cell can divide and become two cells, it has to copy its nucleoid. When it copies its nucleoid, that's DNA replication. What's the process of DNA replication called?
Semi-conservative.
Good, semi-conservative replication. So this way the daughter cells come out genetically identical, but each one has one original strand of DNA and one newly copied strand. It's the only way to be fair. Okey-dokey. Don't forget about your microscopy. Your total magnification, how to calculate it. You gotta multiply the ocular magnification, which is 10 times, by the objective magnification to get your total magnification. That's how it is with a compound microscope. That's why they call it a compound microscope, because you have more than one system of magnification. You have to multiply them together. You know the difference between bright field, dark field, and phase contrast microscopy, right? What kind of microscopes do we have upstairs? Monocular or binocular? Binocular, right, you use both eyes. What's the little piece in the eyepieces that you can turn to adjust for each eyepiece separately? Diopter ring, good.
Okay.
What's the condenser do? Yeah, it condenses. Takes the light from the lamp and condenses it, concentrates it into a finer, stronger beam to go up into your objective lens. And what is the little thing, like a shutter of a camera, inside the condenser?
Iris diaphragm.
Iris diaphragm. Also helps you adjust the amount of light passing through your specimen into the objective. Alrighty. Okay, if I take an antibiotic, what kind of organisms might I be able to get rid of in my body?
Foreign antigens.
Bacteria. Antibiotics will work on bacteria. Some antibiotics work on different things. I just read something recently where they used an antibiotic for something, and I was like, it had nothing to do with infectious disease, but somehow the antibiotic helped with it. I don't remember what it was now. So yeah, the antibiotics are used in different unusual ways. That's why they give antibiotics to cattle also, not just to kill germs and stuff, but it makes them grow bigger and stronger for some reason. We don't thoroughly understand. All right, is an antibiotic going to work on a virus?
No. Nope.
No. Is an antibiotic going to work on an endotoxin? No. Is an antibiotic going to work on an exotoxin?
Yes.
Probably not. But you can make a vaccine for an exotoxin because it's a protein. So an antibiotic won't work on a protein.
Oh, okay, that was gram-positive.
It will work on the gram-positive bacteria, kill them, but it won't get rid of the exotoxin. And if you kill your gram-negative bacteria, too many too fast, what happens to your patient? He has gram-negative infection, and you give him a big dose of antibiotics, wipes out a lot of gram-negatives all at the same time.
Well, intoxication.
What happens is all those dead gram-negatives, that's when their endotoxins are released, when they die. So the patient's going to get too much endotoxin in their system. That can send them into chills, fever, sweats, and even shock. It can even cause your patient to go into shock. So when they have a gram-negative infection, you have to handle the administration of the antibiotic carefully, because if you kill too many gram-negatives at once, then the endotoxin becomes the problem. And the endotoxin, your body gives non-specific symptoms, like fever and inflammation, but one of your non-specific symptoms is if it's too much, the patient goes into shock. And exotoxins. Exotoxins are excreted by gram-positive bacteria, And it could be gram-positive bacteria growing in your vajayjay, which actually is a vajayjay. I don't know why they say J when it's a G. And those gram-positive bacteria, if they overgrow because of that tampon use, they excrete exotoxins into your vagina, and then the mucosal lining, it's very easy for it to be absorbed into your bloodstream. And so that causes toxic shock syndrome. That's why they call it toxic shock syndrome, because it's gram-positive exotoxins that cause it. Now, there's different types of bacteria that can cause toxic shock syndrome, but what are the two most common ones?
Staphylococcus.
Staph aureus, and then there's also one, Streptococcus. Streptococcus pyogenes can also cause toxic shock. It can also release toxins. Yeah, and those aren't the only bacteria that can do toxic shock. There's others that can make, all it has to do is release toxins that cause you to have a shock, and do toxic shock. Alrighty, you know the difference between normal commensal flora, your indigenous microbiota, whatever you want to call it, versus transient flora. And you know the difference between pathogens and non-pathogens. And you know what a strain is, genetic variation of a species. And let's see. You know what zoonotic disease is, right? Transmitted between humans and other animals. And you know what a vector-transmitted disease is. Just means it's transmitted by some kind of a vector. Could be an insect, could be a tick, could be a raccoon. It could be typhoid Mary. Typhoid Mary was considered a vector of typhoid fever. And also what a disease reservoir is, that's a source of infection. There might be a drinking water reservoir that's contaminated. There might be a particular food source that's contaminated. There might be typhoid Mary who's contaminated. She's not only considered a vector, she's also considered a reservoir because she harbors the germs in her gallbladder. The germs were in her gallbladder. They think. And causative organism of a disease is one thing. A vector might be something else. A vector isn't always the causative organism of a disease. And what would we call a virus that's transmitted by arthropods? Arbovirus, right. And which bacteria are acid-fast? Good. Which bacteria lack a cell wall? Good. You remember what causes pneumocystic pneumonia?
Oh, it's a bacteria. You mean walking pneumonia?
Pneumocystic pneumonia was the common killer of AIDS patients. Right? I think it's
Say that again.
Pneumocystis hirovecti. It's a protozoan. Or is it a fungi now? They debated back and forth. Okay, now let's talk about walking pneumonia. What causes that? Mycoplasma, right? So walking pneumonia is bacterial. Pneumocystic pneumonia is protozoal. Is it protozoal or fungi on your list? Yeah, it's fungi now. They thought it was protozoal and then they decided it was fungal. Yeah, it's a yeast. It's considered a yeast, like a single-celled fungus. Yeah, so and how about viral pneumonias? Right? And how about gonorrhea? Gonorrhea is Neisseria, but so is meningitis. So sometimes you'll see related bacteria can cause different diseases, and sometimes you'll see different kinds of organisms can cause the same disease. So when a patient has pneumonia, it just means the patient has pneumonia. It doesn't tell you what's causing the pneumonia. There's different types of pneumonias. There's viral, bacterial, fungal, all kinds of stuff. Meningitis also. You get meningitis from gonorrhea, from Neisseria meningitis. You can get meningitis from molds and fungi, and you can get meningitis from bacteria, and you can get it from viruses. So sometimes patients will have a disease, but the causative agent could be different. And sometimes the causative agent can be the same thing, but it results in different diseases. Just want you to keep an open mind about all of that. What causes mononucleosis?
A virus.
What kind?
Epstein-Barr?
Right, Epstein-Barr virus. What family is that in? Herpes viridae. And herpes viridae hides inside a host cell, so it's a latent infection. So what does that mean if your patient has mononucleosis? He's gonna have that in his system forever.
Coronavirus.
Exactly. For a long time, the theory was that mononucleosis would remanifest as chronic fatigue syndrome. So they thought that chronic fatigue syndrome patients, what they were doing was, you know how chickenpox becomes shingles? Well, they were thinking your mono that you had as a teenager, then as an adult, it came back as chronic fatigue syndrome. That was a big theory for a long time. So, yeah, those herpes viruses, all of them, once you've got it in your cells, it's there for your lifetime. You might never have the disease again. Your immune system might deal with it, or it might never be replicated and activated in the host cell. We don't know what causes some people get shingles and some people never do. But when you're immunocompromised, that's when things tend to erupt. But all those herpes viruses, you're gonna have them forever. All right, you know what a bacteriophage is? You know what cytopathic effect is, right? And you know about retroviridae. What's the special enzyme they have that helps them do transcription and
Reverse
reverse the whole cell?
transcriptase.
Right, reverse transcriptase, good. And what kind of virus causes rabies? What family? Sounds like rabies, Retroviridae, good. And